Okay, so this next section of the lecture is going to talk about basically how neurons are set up and ready to pass on these electrical impulses down the neuron. And we'll really kind of break that up basically. And I'm going to break this up into two kind of shorter lectures, mainly for you. So that way, then we're first going to talk about how we actually set up what's called a membrane potential. And that allows a neuron to be in a state where it's potentially excitable, right, at all times. So we're going to talk about how neurons set up that resting membrane potential. And then in the next lecture, we're going to talk about how they actually can transmit that electrical impulse. Okay, and the ways that they do that. So again, this first one's going to focus on the fact that these neurons are these electrically excitable cells. And this is going to be due to something that we call a voltage difference across the membrane. We're going to talk a little bit more about that in just a minute. And that's really what sets up their membrane potential and allows them to be potentially excitable. And then again, in that second part of the lecture, we're going to talk about how they actually communicate with these electrical signals. And there's going to be two types of electrical signals that we'll talk about in that next lecture, one called action potentials that travel long distances, and ones that are called graded potentials that are going to travel shorter distances. So again, that'll be the second lecture that we'll talk about that second part. So this lecture is just going to focus on this first part here. I can get my slide to advance here. So again, we're really setting ourselves up to understand this kind of whole greater process of how we can actually do something like, again, our fine motor skills with writing with a pen or pencil. And so again, you have to have things like these, oops, if I can get there we go, my mouse, these sensory receptors here that are going to be in the fingers, the touch receptors and the pressure receptors and things like that are going to be sending information back, right, to the brain through the spinal cord. And they're going to be sending this information back to the brain so that, again, in the integrative centers in the brain, we can then decide, again, make decisions upon what motor output is needed. Okay, so, and then here you can see here, again, signals are going to be sent along these motor neurons and out to the muscles in the hand to allow those fine motor skills that allow you to write. So it's, again, kind of amazing that all of this can occur in just split seconds. And so we're going to talk about how that is and why that can happen so quickly. And we're going to really focus here on how we can, in this kind of next couple lectures, and how we can actually transmit these impulses, these messages through electrical impulses down these neurons. Okay. And so when we talk about this, when we're, again, transmitting these electrical impulses, remember that we're going to have these neurons that are, again, taking in information through those dendrites. So again, passing electrical impulses or stimulating an electrical impulse, again, in these dendrites. And then passing, again, that electrical impulse down the axon and out the axon terminals, okay, to, again, either another neuron or to your effector, like your muscles. So again, that's really what we're going to be kind of focusing on. And to really do that, you need to kind of go back to that one of the first lectures that we had and kind of remember the different types of ion channels that we have in the membrane. Because remember that ions cannot easily cross the membrane. So remember, things that are large and charged don't just cross that phospholipid bilayer. So there's channels, and we have these different types of ion channels. So we're going to have these two types of ion channels. We have leakage channels, and these leakage channels are kind of like what you picture here, water kind of leaking out of a faucet, just like it's kind of described. They're always open, and it just allows, and it's usually specific to a certain ion, and then it just allows that ion to just kind of leak through. So your neurons have both potassium and sodium leak channels. Again, some of those leak channels are specific to leaking potassium out, and some of them are specific to leaking sodium out. And our neurons are always going to have more potassium leak channels than sodium leak channels. And I'll explain why here. This will make a little more sense in a few minutes here. So what this does is just kind of keep this in the back of your brain that the membrane is actually going to be more permeable to potassium than it is to sodium. Okay. And this is really going to eventually help us explain how we set up the membrane potential across, again, your axon membrane. So this will, again, be kind of important, but we're going to come back to this in just a minute. So those, again, our leakage channels are one type of channel. And then our other type of channel are called gated channels. And these open and close in response to certain stimuli. And we're going to talk about there's different gated channels. Some of them, again, are going to respond to like a chemical stimulus where they bind a ligand. Some of them will respond to an electrical stimulus where there's like a change in electrical potential that stimulates that gate to open on that channel. Okay, so again, we've got leakage channels and we've got gated channels here. And so, here's my slide on the types of different gated channels. So we have these 3 different types of gated channels. We have chemically gated channels. Sometimes they're also called ligand gated channels because, again, remember the binding of that chemical to a receptor makes that chemical a ligand. So both names are appropriately used. So again, these chemically gated channels just require the binding of a certain type of chemical to open up that channel. So we already kind of talked about this when we talked about neuromuscular junctions, right? So we had those, remember the acetylcholine receptors? And those acetylcholine receptors, right, once they bound the acetylcholine on the motor end plate, it stimulated a sodium-gated channel to open, okay? So again, thankfully, we've kind of already seen an example of that, but we're going to see more examples of those chemically gated channels coming up here with different neurotransmitters especially. So again, chemically gated binds a chemical to open up that gate. Voltage gated channels, we're going to talk about what voltage really is here in just a minute, but these are going to be channels that are going to respond to these changes in electrical potential across the membrane. So change in voltage across that membrane. And I'm going to talk about what voltage is here in a little bit more detail in just a minute for those that don't really know. And then we have these mechanically gated channels. And these we're typically going to see in like sensory receptors. These are going to be ones that are going to open and close in response to some type of mechanical change in that receptor. So for example, all your touch receptors and your pressure receptors, okay, those are going to be stimulated with these mechanically gated channels. You are physically causing a physical change to that receptor by, pushing on it or, again, touching it. So these are all different types of channels that we'll kind of talk about, and we'll talk about their role that they play throughout these next series of lectures. So just real quick, when you talk about the chemically gated, and we're going to kind of focus on chemically gated and voltage gated ion channels because they're going to really help us kind of understand the resting membrane potential and the action potential here in the beginning. So again, our chemically gated channels just here bind a receptor, and then once they bind that receptor, it causes, again, that physical change within that protein channel, allowing it to open and allowing things like sodium to come in and potassium to come out. Now remember, we kind of talked about there's this concentration gradient that we set up across the cell membrane with these sodium potassium ATP pumps, okay? So we're gonna constantly, those sodium potassium ATP pumps are constantly pumping out 3 sodium per 2 potassium. And what that means is that we're always going to have this buildup of sodium outside the cell and potassium inside the cell. So anytime that we open these gated channels, that means that sodium's going to want to rush in and potassium wants to go out. Okay. So again, I'm going to talk, I have a slide on this to remind you of that for the sodium potassium ATP pumps, but I just kind of wanted to bring it up now too. And then we have, again, so we've got the chemically gated ion channels where it just, again, binds a chemical, oftentimes a neurotransmitter, to open up that channel. And a lot of times that channel is going to allow sodium to rush in. And our voltage gated ion channels are going to open in response to a change in membrane potential. Okay, so basically a voltage change, again, which we're going to talk about in a minute, and that causes that protein channel to open, allowing sodium to rush in. So when, again, our gated channels are open, the ions can diffuse really quickly. They're going to diffuse oftentimes, again, down their concentration gradients. So again, like we talked about with sodium having a higher concentration outside the cell and potassium, higher concentration inside the cell, they're just going to, as soon as that gated channel opens, they're going to diffuse right down their concentration gradients. And then the electrical gradients are just going to mean that things are going to want to move towards the opposite charge. So in a little bit, we'll talk about when you have a buildup of a negative charge on the inside of the cell, then again, any of the positively charged ions are going to be attracted to those negatively charged ions in the inside of the cell, right? Because opposites attract. And so sometimes, again, that change in that electrical creates an electrical gradient when you have a buildup of positive charges on the outside of the cell and buildup of negative charges right on the inside of the membrane. Okay, so oftentimes these two work together, and because, again, here we have sodium that wants to move to the inside of the cell down its concentration gradient, but it also wants to move to the inside of the cell because of this electrical gradient that we're going to talk about. Okay, and together we call that the electrochemical gradient. Okay, so again, kind of keep this all in mind. I know I'm giving these like little pieces to the puzzle, and it will all fit together and the very end. So there's just lots of little pieces to this puzzle. And this is just kind of showing you the same thing. I just like this depiction of these gated channels, this voltage gated channel. And you see, again, that change in voltage across the membrane. And then it kind of shows you the gate actually opening up here. So I liked that depiction of that. But this is just, again, the same thing showing you voltage gated ion channels and ligand gated ion channels or chemical gated ion channels. Okay. So let's go back and let's talk about some of these basic principles of electricity. So sorry about that. My computer just shut off and turned back on. So hopefully it doesn't create too much of a jump in here. But anyway, so we're going to be talking about some definitions for talking about these basic principles of electricity, right? So it's important to understand what voltage is. So voltage is literally just a measure of potential energy that's generated by a separation of charge. And across the membrane, across the cell membrane, is where we're going to see this separation of charge in our neurons. Now, again, you kind of probably intuitively know this because you've heard the word voltage in terms of batteries, right? Although usually it's in like a 9-volt battery. Now here, when we're talking about the voltage across a membrane, voltage change across a membrane, it's going to be in millivolts. So we're talking about a millivolt, again, 1/1000th of a volt. But again, still enough to do the job that we need it to do. So again, just like a battery is going to have that separation of charge, positive and negative ends, same here in terms of our voltage change, but it's just going to be across the membrane. And so again, when we talk about voltage, it's just a measure of that potential energy due to that separation of charge, okay? And then that voltage then can lead to the flow of that electrical charge from one point to another. And that's what we call current. So again, when we have a separation of charge across the membrane, positive on, one side, negative, maybe positive on the outside, negative on the inside. And then the current is going to be when we open up one of those channels and allow the flow of ions. Okay, so again, those positive ions are typically going to be attracted again to those negative ions, right? Opposites attract. And that's what current is that the flow of that electrical charge. Okay, so, and this is just kind of my funny slide for you to remember that again. Current is that flow. Voltage sets you up. set you up for this, but current is actually the killer here because current is the flow, right, of that electrical charge. Okay. And I really like this diagram of showing you, these electrons wanting to move. And here's the voltage is kind of pushing it along, right? So again, I just like this idea of like voltage is really the push to set you up for this current, right? For that move of that electrical charge. So that's again what's happening with resting membrane potential. Resting membrane is going to set us up with that voltage, right? And so again, much like a battery, where we've got, again, the separation of charge on a battery, we're going to set up that separation of charge across a cell membrane. And what's going to end up happening, and we'll talk about how we do this in just a minute, but we're going to end up with a more negative charge on the inside of the cell membrane and more positive ions kind of lining the outside of the membrane, okay? And so it's this separation of charge across the membrane that actually it sets up our voltage, right, across the membrane, because again, the voltage is that separation of charge. So you can actually measure this. They actually have these little tiny electrodes, believe it or not, like again, microscopic electrodes that they can stick into a cell membrane, and they can actually record this this voltage change across the membrane, what we call our membrane potential. And typically, this voltage change across the membrane is usually around negative 70 millivolts. That's, again, the measure of that potential energy difference between the positive and the negative charges in the inside of the cell versus the outside. And so again, this is what we call a polarized cell. And try to keep in mind that in a neuron, right, this would be like the axon. So here, if you have it, let me find my color here. So again, here would be, one part of one side of the axon, and then down here would be the other side of the axon here. And on the other side of the axon, it's going to be the same thing, right? So you're going to have more negatively charged ions lining the inside of the axonal membrane here. And then you're going to have more positively charged ions lining the outside of the membrane, okay? And so you'll see this all down the axon. Again, and this is what we call our resting membrane potential. Typically, again, resting membrane potentials, it's good to know this number, resting membrane potential is around negative 70 millivolts. That's that potential difference that we're going to find in our, again, neurons at rest, okay? So let's get farther into this. Let's talk about how we get this, and that really hopefully will help you to make sense of this, how we actually got this resting membrane potential. So, and then again, this is just kind of showing you the same thing where they're using this microelectrode to measure that voltage difference. Pretty cool. So the three factors that are going to lead up to this resting membrane potential are, one, and this is the most important one, is going to be the unequal distribution of ions in the extracellular fluid versus the cytosol. So again, this is outside the cell. You can put outside the cell versus inside the cell, okay? And so the extracellular, let me see if I can kind of draw this for you here. So again, here is our axon, and here's, oops, if I can draw this here. Oh, I did not mean to do that there. But here is going to be our membrane. Oh gosh, my drawing is to be desired here. Oops, hold on. Let me get this back here for 1 second. So there we go. Sorry about that, guys. Not sure what happened there, but again, let me kind of draw. Here is your, again, axon, okay? So here's going to be inside the cell, and this is out, okay? And so again, we're going to build up because of these sodium potassium ATP pumps, okay? Remember, these sodium potassium ATP pumps are constantly pumping 3 sodium out, okay? so lots of sodium out, per every 2 potassium into the cell, okay? And this is going to create this imbalance here of the sodium and potassium, okay? So you're going to have lots of potassium inside the cell, because then, again, it's going to pump 3 sodium out, but then it's going to pump 2 potassium back in, okay? So we're going to end up building up this kind of concentration gradient of sodium outside the cell, and it really kind of wants to move back down as concentration gradient into the cell, and then potassium inside the cell, okay? Now, keep this in mind that there are going to be more potassium, just like we talked about, more potassium leak channels than there are sodium. And this is the most important factor in membrane potential. And so what's going to happen is potassium is just going to kind of continually, kind of like a leaky faucet. There's going to be these certain leak channels that are very specific to potassium. And they're just going to kind of constantly leak potassium out of the cell. Now, keep in mind that, again, potassium is a positively charged ion. So what happens is as that potassium leaks out of the cell, you're going to start to get a buildup of a positively charged ions, right? Because you're leaking out much more potassium than sodium is back in. So, because there are some sodium leak channels, but they're much fewer, okay? And so, again, this is going to allow this positive charge to kind of build up on the outside of the cell membrane. Now, what's going to happen is there's all kinds of negatively charged ions inside the cell, again, just naturally in your cytosol. And what's going to happen is those negatively charged ions are actually going to be attracted to these positive charges of the potassium being leaked out. And so you're going to build up right against the cell membrane a bunch of negatively charged ions. Again, they're just attracted, right? Because opposites attracted to that positive charge, but they can't move across because there isn't a channel allowing them to do that. So again, they just kind of build up that charge at the membrane. And this is really the most important factor in setting us up for this voltage difference across the membrane, this membrane potential, okay? So again, really important to understand that. Now, again, the second factor is, of course, again, the inability of most anions. Remember, anions are negatively charged ions. So the inability of these negatively charged ions to be able to leave the cell, okay? So again, most of the negatively charged ions are not free to leave the cell. So they're stuck inside the cell. And that's why, again, they're going to kind of build up at the membrane as they're attracted to those positively charged potassium ions that are there. And then, again, the third one is, of course, this sodium-potassium ATP pump. Okay, so remember, this does require ATP because we're pumping against a concentration gradient, right? We're pumping sodium out, even though we're building up sodium outside the cell, and then we're pumping sodium back in, again, even though it wants to leak down. But again, remember that we're gonna have, even though we have these potassium leakage channels, okay, we're still pumping in more potassium than is leaking out, okay? So we still have this nice concentration gradient. And so again, for every 3 sodium, okay, that are pumped out, only 2 potassium are pumped back in. So again, we're pumping out 3 positively charged ions and only 2 back in. So that's just another factor for helping us set up that membrane potential. It helps to contribute to that membrane potential, but most importantly, right, it's setting us up with this concentration gradient so that, again, potassium can leak down those leakage channels. And again, this plays the most important role there. And I like this. There are 25 times, the membrane is 25 times more permeable to potassium than it is to sodium. So again, that's why we leak so much potassium out. So that set us up here with our membrane potential, okay? Now let me kind of show you some actual images of this. Okay, so again, here you can see they're just showing you that potassium leaking across, right? And so what ends up happening is so much potassium actually leaks across that the cell interior actually becomes quite negative. It's at a negative 90 millivolts because again, all of these positive charges are leaving, and that leaves us with a buildup of negative charges. So the inside of the cell actually ends up at negative 90 millivolts. Now again, remember that we have some sodium leakage channels that allow some sodium back in, and so they bring in a little bit of positive charge back into the cell, and that ends up leaving us at right around 70 millivolts as we allow some of those positively charged sodium back in. Okay, so that ends up again giving us this overall resting membrane potential of about negative 70 millivolts. Okay, and then again, this is just kind of reminding you that we have to have this sodium potassium ATP pump, right, to be able to keep our concentration gradients of sodium and potassium. Okay, so again, we're going to constantly pump 3 sodium out per every 2 in. Okay, remember that this is what we call that symporter. So again, it does both. It's going to again bind sodium and pump it out, and then bind potassium and pump it back in. Okay, so that's that symporter. Okay, so that's just kind of a diagram. I like the visuals of all of that. And again, just showing you the same thing here, pumping sodium out. So we get that buildup of sodium, and then pumping potassium back in. I just liked this one because it actually shows you this concentration gradient. So you can see, again, you have a lot more potassium inside the cell than you do out. So you can see how potassium wants to move down that concentration gradient. And then here, same with sodium. A lot more sodium outside the cell than inside the cell. So you can see how sodium wants to move down that concentration gradient. Okay. And then here they're just showing you again the buildup of charges. Okay. So again, and this buildup of charge is going to be right along the membrane because again, as those positively charged potassiums leak out and kind of are hanging out right out here, right outside the membrane, then these negatively charged ions inside the cell are going to be attracted to all those positive charges, even though, again, they can't move through that membrane because there isn't a channel for them to do so. So this is a great little flick. I'm just going to play it for you really quick because
the human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts.
Potassium was the only ion moving.
Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks.
And raises the voltmeter reading to minus 70 millivolts.
Thus, the pumps help to maintain the resting membrane potential.
Potassium pumps actively transport sodium ions out of the neuron and potassium
So
ions
again, you can
back
add
in,
animation
compensating
from
for the sodium
this
and
lecture when you
potassium
open it up.
leaks.
It's a great animation. I love it because it really helps to
Thus,
put
the
everything
pumps
together.
help to maintain
So
the resting
play that
membrane
as many
potential.
times as you need to. That's why I put it in the lecture. So the next section of the lecture is going to talk about how we change the resting membrane potential and kind of why we change the resting membrane potential in terms of, again, transmitting these electrical impulses down the axon. Okay, so as always, please let me know if you have any questions or concerns. Again, I'm here to help.